We start with Colloms from University of Glasgow. Thank you.
Okay, thank you very much, Andre. My microphone's working? Yep. Okay, so this morning I'm going to be telling you about recombination, XCR site-specific recombination at plasmid sites. So this is work that I started at the University of Oxford with Dave Sherritt, where I met Ian, who's talking next, and I've carried on at the University of Glasgow. And Andrew Travers doesn't seem to be here in the audience this morning, and he probably won't remember this, but he actually introduced me to this field in Cambridge back here in about 1987 to the idea of DNA bending, DNA binding to nucleosomes. And somebody who shall remain nameless noticed a slight family resemblance, they thought, to this gentleman who was staring down at us at dinner time on Wednesday night. Right, so I'm going to start my talk by giving you a brief introduction. So at this point, I'm a biologist, but I'm going to tell you about the biology of these topological changes, but I hope I'll make it simple enough for the mathematicians to understand as well. So I'll start by giving you all an introduction to what is site-specific recombination and why it causes topological changes. I'll give you a one-slide introduction to what is this XCR recombination system. And then I'll tell you about, well, mainly catenation, that knotting and catenation changes that take place during XCR recombination. And if time allows, I'll go on to tell you about some linkage change experiments, so change in the linking number during XCR recombination. So site-specific recombination takes place at short DNA sequences, 30 to 40 base pair long core sequences, to which recombinase proteins bind. So these sequences have slight inverted repeat sequence symmetry, so they're dyad repeats, so they can bind to a dimer of recombinase protein, and then two dimer-bound sites can be brought together by a tetramer of the recombinase protein. And once this tetramer has formed, the recombinase protein itself cleaves both sites, and for the family of recombinases I'm choosing to illustrate this with here, they use a serine residue, and the tetramer cuts all four strands of the two participating sites. Once the two sites have been cut, one site can start rotating with respect to the other, all the way around through 180 degrees, and then the sites can be religated in this new recombinant configuration. So if you imagine that these ends are connected off to a circle, this crossing here, will change the topology of that plasmid circle, that DNA circle. So each of these is a double-stranded DNA molecule. These short core sites tend to have some asymmetry in them so that they have recognizable left and right halves, and a left half is almost always joined to a right half during the recombination reaction. And so that means depending on how the two sites are arranged in the DNA molecule, you get different kinds of DNA rearrangements. So if the two sites are pointing towards each other, like over here, when you cut both sites and rejoin the pointy bit of this site to the square bit of this site and vice versa, we end up with this, which is inverted the orientation of the segment in the middle. If, on the other hand, the two sites are pointing in the same direction in the DNA molecule, we get a deletion reaction. So if we start with one circle, we end up with two circles. And the reverse of that reaction is a fusion reaction. So one circle can integrate into another circle. Right, so that's that's site-specific recombination for you. This is my one-slide introduction to the biology of the system that I work on. And so these circles up here represent plasmid molecules that are free-floating circles of DNA that replicate inside bacterial cells. And there are processes in these cells called homologous recombination that can take two of these circles and join them together to make a circle that's twice as big, a dimer. And these dimers can end up taking over single cells in a growing population of bacteria. And the dimers are maintained in cells at a lower copy number per cell than the original monomeric plasmid. And at cell division, the plasmid molecules, individual plasmid molecules, or maybe clumps of plasmid molecules, segregate at random to two daughter cells. And so the fewer copies of plasmid you have in the cell, the greater the chance that a cell division will produce a cell with no plasmid copies in it. And the plasmid is essentially a selfish piece of DNA. It wants to survive in the cell. And so many plasmids have systems to make sure that they never get into this situation. They never become dimeric. And so the plasmids I'm going to talk about today have special sites on them, recombinase sites that are either called the Xer site for Colli1 resolution site, or the Psi site for PSC 101 something or other, something French, I think. And so the names just reflect the original plasmids, natural plasmids they were found on. But so these sites are recognized by proteins produced by the bacterial cell, recombinases produced by the bacterial cell, and recombined to form monomeric plasmids. Okay, so the system is regulated, though, so that recombination only occurs if you have two sites facing the same direction in actually a supercoiled DNA molecule. So you need supercoiling, and you need two sites, and you need them to be facing the same direction, direct repeat in a circle. If they're an inverted repeat, they don't recombine, and two sites on separate molecules don't recombine. And so what we were interested in is how does this regulation work? How can these sites recognize that they're on the same molecule, pointing in the same direction, and only recombine then? So if we look at the structure of one of these recombination sites, we see it has a 30 base pair recombination site that we call the core site. This is like in my cartoon in my introduction. This is the site where DNA strands are broken and cleaved by recombinase molecules, recombinase proteins. But there's also an additional roughly 180 base pairs of DNA that's required for recombination. And you might hear me refer to this extra sequence as the accessory sequences. So the accessory sequences are required for recombination, and I'm supposed to have moved on to my next slide here. There are also two proteins that are required for recombination that bind to these DNA accessory sequences. And they are the ARG-R protein, which is a DNA binding protein that has some other role in the cell that regulates gene transcription, but it binds specifically to an 18 base pair sequence in the Xer accessory sequences. And the other protein that's involved in this recombination reaction is a large hexameric peptidase protein that also just so happens to be another DNA binding protein. So we'll go on eventually to see a model for maybe how Pepe can bind to DNA later in the cell. So if we look at most recombination reactions carried out by site-specific recombinases, we find that if you give them a supercoiled DNA molecule as a substrate, and as Andre said, you can do this with purified protein in the test tube. You just add a recombinase to this. It will recognize the two recombination sites and bring them together and recombine them to make two circles. But these two circles won't be two separate circles. They'll tend to be joined together like this in a link or a catenane. And because you can bring these two sites together with different numbers of supercoils trapped between them, you get links with different topologies. So I've illustrated that here with the lambda integrase system, a system that just brings sites together at random and introduces random numbers of links in the catenanes that it produces. And so we can see that here. So what we've done here is we've run a gel. So we've done a recombination reaction. We've actually nicked the DNA to get rid of all supercoiling because that interferes with looking at how knotted the DNA is. And then we've run a gel, and these gels separate knots and catenanes according to how many nodes or crossings they have. So the more crossings they have, the more compact they are, and the faster they run in the gel. And so lambda integrase produces 4-noded catenane, 6-noded catenane, 8-noded catenane, 10, 12-noded catenanes. And that's because of this random collision of the sites trapping random numbers of supercoils. In contrast, the XCR site-specific recombination system produces a catenane, this 4-noded catenane with a specific topology. So it's this 4-noded catenane with the 2 recombination sites in anti-parallel around the catenane. And so these 2 bands that we see here are actually small amounts of the circles that are released by me being too vicious with the nicking enzyme that I need to use to get rid of the supercoiling so we can see this. Okay. So I told you from that gel there that this is a 4-noded catenane, but how do we really know it's a 4-noded catenane? Well, one way of checking this is to run the product of recombination next to a ladder of knots produced by topoisomerase 2. And topoisomerase 2 produces twist knots that are the 3-noded knot, the 4-noded knot, 5, 6, 7-noded knot. And if we start with a DNA molecule that's the same size as the DNA molecule that I've done that has the XCR recombination sites on it, so this is the unknotted circle. These then are the knots made by topoisomerase 2, and the catenane made by the XCR recombination system runs at the same place as this 4-noded twist knot. So that tells us that it's a species that has 4 crossings in it. We can analyze it in other ways. We can do restriction digest and show that it is actually composed of 2 circles. But another thing we can do, or attempt to do in my case, is coat the DNA with Rec A and do electron microscopy, and then you can see some of the molecules you see that haven't been ripped apart by the nasty treatment that I had to do this are you can see are 2 circles, and at least for some of these crossings you can see which DNA segment is going over which other one. And who did we say the old-fashioned way of doing it was you would just uncover 1 crossing at a time and have a vote to decide whether this was an over or an under. And I think probably to assign every one of these crossings you would need better pictures than the pictures I was getting. But we also had other ways of actually showing that it was a right-handed catenane like this with 4 crossings in it. Okay, so in summary of this little section of the talk, the XCR recombination system makes this specific 4-noded catenane, whereas other systems tend to make families of more complex knots and catenanes. So how do we think this is happening? Well, our model is that the Pepe and the RGR proteins are binding to the accessory sequences and wrapping them around each other in this right-handed plectonemic sense, trapping exactly 3 supercoils. And this then brings the core sites together, these green segments, for strand exchange for cutting and rejoining reactions by the recombinase molecules. So if we show that in a slightly different representation, these blue sequences here are the accessory sequences, interact a precise number of times around the Pepe and RGR proteins. That brings the core sequences together for recombination. And after cutting and joining these, we end up with this, which is the 4-crossing catenane. And if we say formation of this complex is essential to allow strand exchange, and formation of this complex can only occur between sites that are directly repeated in a supercoiled molecule, formation of the same complex would be unfavorable between sites in separate molecules, then we get this topological specificity recombination only when the sites are in the same molecule in direct repeat. Okay. And so the model accounts for the requirements for the accessory sequences in the proteins, the fixed topology of the product, and this intramolecular selectivity. Okay, so a few years back, Marielle Vasquez, who's in the audience here, used some maths to show that In fact, there are, given some simplifying assumptions, but very biologically reasonable assumptions, there are 3 Tangle solutions that would give you this 4-noded link, this 4-noded catenane as product. And one is essentially the one I showed you in the previous slide, with 3 crossings trapped by the accessory sequences. There's another possibility with 5 crossings trapped by the accessory sequences. And for these 2 to work, you just have to have a different sign for the crossing that's introduced by the recombinase. And the other possibility is to have 4 crossings trapped by accessory sequences, wrapping around the accessory sequences, and the sites to come together, the actual core recombination sites to come together in an anti-parallel way, so that they flip from being like this to being like this, and the product, again, is the 4-noded catenane. So, mainly because earlier in the week we were talking about protein knots and computer programs to take a knot in a protein and to close it by joining the ends together in different ways, just on the computer, I thought I would show you my experiments where I've actually done this in a test tube with a DNA molecule. So the experiment is to take a DNA molecule, for which this is going to have to suffice, and the DNA molecule has 2 recombination sites in it. So we just take this DNA molecule and add the recombinase proteins, or at least the accessory proteins RGR and PEP, and then the sites, the 2 recombination sites, will wrap around each other with these 3 or 4 or 5 crossings, and then we add ligase and join. I needed DeWitt's rubber tubing for this. I nearly stole it yesterday from his flat, but so we take the 2 ends and join them together, and we get a knot, which in this case, I think, if I did that right, would be a 5-crossing knot, probably this twist knot here, if I did what I draw on here. So we've formed the complex, and then joined the 2 ends together, and then at some probability, we get knots. So if we look at this gel, if we just ligate a linear in the absence of any proteins at all, we get a lot of unknotted circle and a small amount of the 3-crossing, the trefoil knot. If we add RGR and PEP together to the DNA before doing this ligation, we still get some unknot, but we get a significant amount of this 5-noded twist knot. And if we just add the recombinases as well as RGR and PEP, we still get lots of knots, but the distribution of knots has changed, and we're getting more of this 6-crossing knot. And so what I'd love the mathematicians to be able to do, but I'm sure they'll all tell me it's impossible, is to go, instead of going from a knot that you've computed in your computer, or an open knot and closing it, and seeing what distribution of knots you get, I would like them to be able to go from this distribution of knots that I'm obtaining in practice and go backwards and compute for me the exact structure of this. But they'll all be shaking their heads now. But that's a problem they can go away and work on. So we have crystal structures for RGR and PEP, and we've done a number of experiments to try and find residues on the surface of this. So this is a hexamer of the PEP protein, another hexamer of the PEP protein, and a hexamer of the RGR protein here in red. And a few years ago, we did a whole series of experiments looking for mutants in PEP that abolished DNA binding. And so I've shown them on the surface of the PEP molecule as these red blobs here. And they tend to form lines along the surface of the PEP molecule of positively charged amino acids. So mutants that abolish DNA binding fall in these positions. And so what you can do from the location of these mutants is trace or thread DNA along the surface of PEP, following as much as you can anyway these lines of residues that are important for DNA binding. And you can come up with, so this is a pure modeling exercise, this thing isn't real, the structures that it's made from are real, but this massive monster thing, my monster synapse as I call it, is a feat of imagination, triumph of imagination. but the DNA has been threaded along positions that we know are important for DNA binding. And also, I wonder if we can just, oops. Can I make the movie go again? They also go through this Star Wars-like groove that's in the Pepe hexamer, like no, can't remember. I was going to play some music to this, but it was technically beyond me. But you can imagine in the Death Star that canyon of death thing and all the right. Okay, so in the last however long I've got for my talk, I've got about 10 minutes. Oh, okay, so I can relax. Yeah, okay, so I'm going to tell you about experiments to try to determine the linkage change that takes place during the Xer recombination reaction. Okay, so first I better tell you what I mean by the linkage change. So if we think of a circular covalently closed DNA molecule with two recombination sites in it that are directly repeated, and this DNA molecule will probably be supercoiled as well, and we add recombinases to it in vitro and we get two products. And to make the picture easier to look at, I've shown them as separate circles, but remember they will be linked together in this 4-cross concatenate. So the linkage change of the reaction, which I'll be calling delta LK, is simply the total linkage of the two strands of DNA around each other in this product, plus the total linkage in this product, minus the original linkage of the substrate. Is that what I've written there? Yeah. So the problem with that is it's hard, very hard, to determine the absolute linkage in a DNA molecule like this. This is typically a 4,000 base pair DNA molecule. So if it's 4,000 base pairs long and it has just these 8 negative supercoils that I've drawn in it, it's going to have an absolute linkage of the two strands around each other of something like 370, 372 I've calculated. And the two products will be about 187 each. And so that would give a linkage change of +2, if my arithmetic is correct. But it's very difficult to determine the absolute linkage of a DNA molecule like this. But what's much easier to do is to determine the linkage difference of a DNA molecule. In other words, how supercoiled it is, which is the absolute linkage minus the linkage it would have if it was relaxed, LK0. So the linkage difference, LK minus LK0 for the product, the two products, and the substrates should be measurable. And what comes next? So I've just illustrated this. We might be able to measure that our substrate has 8 negative supercoils in it, and we get two product circles, each with 3 negative supercoils in it. That would give us a linkage change for the reaction of +2. So why would we want to do this? Well, not really for the first two reasons, although they may have had a small part to play. But it's because it can provide us with information about the actual mechanism by which the strands are cut and rejoined during the recombination reaction. And so I'm just going to illustrate this to you with 2 examples so we can dream up 2 possible reaction mechanisms by which the strands could be cut and rejoined at the core sites during the recombination reaction, and illustrate that they end up giving you different linkage changes. So you could distinguish, therefore, between different models for the strand exchange reaction if you can determine the linkage change. So this is the first model I'm going to illustrate to you, which is called a simple rotation model. And it's thought to be used by some recombinases. So if this is 2 recombination core sites brought together by a tetramer of the protein, all 4 strands get cut at once, and then you simply rotate 1/2 with respect to the other. And that introduces this crossing between the 2 sites, but it also introduces a twist in this DNA molecule and a twist in this DNA molecule. So just to illustrate this, if we imagine starting with a substrate that only has 4 negative supercoils in it, three of them are these three crossings that are going to be trapped by Pepe and RJAR wrapping accessory sequences. And just to make the strands end up in pretty positions, I've put a half twist and a half twist in up here and up here. And if we do this simple rotation mechanism, we end up making the four-noded catenane by introducing this cross here. So we end up going over, under 4 times, making the four-noded catenane. And we also introduced an extra negative half twist here and here by doing that rotation mechanism. And what you have to imagine doing is cutting one of these circles away and looking at the linkage left in the other circle. And if you do that, you find that this guy has -1 and this guy has -1 supercoil. So we go from our original substrate that had -4 supercoils in it to a product that, if you add up the linkage in the two circles, adds up to -2, so the linkage change is plus 2. And we can do the same thing for a different type of mechanism, one in which the two sites are brought together in anti-parallel. We cut only one strand in each site, so strand here, and then rejoin them to make this holiday junction intermediate, and then we cut this strand here and this strand here, and end up having made the full recombinant. And if you plug that mechanism into something that gives us the right four-noded catenane at the end, the linkage change ends up being +4. So if we can measure the linkage change, we could distinguish, for instance, between these two models. So the first way I tried doing this was to start with a DNA molecule, plasmid, supercoil circular plasmid, roughly 3.3 kb, and put two sites close together in it so that when it recombines, it gives a large circle and a small circle, a 300 base pair circle. And this circle will probably only form one topoisomer. The energy difference between topoisomers will be sufficiently different that its thermal variations won't give you the different topoisomers. So we should get one specific topoisomer product of this, and therefore one specific topoisomer product of this, if there's a fixed linkage change for the reaction. So now we get to the pretty gels. So this is a two-dimensional gel. Don't worry, I'm going to try to take you through this. But the main thing to remember is that this type of gel can separate DNA molecules of the same size just according to how many supercoils they have in them, how many units of twist is in them. So for instance, maybe the other thing to remember is that the slowest topoisomer is the one with no supercoils in it, LK minus LK naught of 0. So this is a gel that's actually been run in 2 dimensions, but if we just think of this second dimension where there was no intercalator in the gel, for this plasmid is the slowest topoisomer and therefore has LK minus LK naught of 0. And then this has just one more unit of twist in it. The DNA, you could imagine taking a circle of DNA, breaking both strands, just rotating by 360 degrees, ligating again, and we get something that migrates at a different position in the gel. So that's the -1 topoisomer, the -2, -3. You can trace it all the way around there. So this is one plasmid, an arc of plasmids, topoisomers that are all the same size but have different numbers of linkage in it. So that's actually a ladder I made for my large 3.3 kb substrate. I've made an equivalent ladder which goes around here, and I think we should just about follow it there and down there, of the product, the large product circle. Okay, and so just to make it easier to see where I think all these bands are, I've marked in red the substrate topoisomer ladder and the product topoisomer ladder, and you can count from this 0 all the way around individual bands that each differ from each other by 1 linkage unit. And so then what I've done is I've taken 4 different purified topoisomers that I've just run on the gel here so that we can count. So this one here corresponds to that one there on the ladder, this one to that one, etc. And then in another lane, I've taken those and recombined them and then cut away the small circle so that we can look at the big circle product, and we get, and I'm afraid they were quite faint on the gel, but I assure you they were here, these 4 different products from these 4 different topoisomer substrates. So this guy goes to that one, to that one, etc. And if you feel like checking my counting, I allege that this one is topoisomer minus 19, starting from 0 over there, and that the product you get from that is topoisomer minus 14, again counting from 0 over there. And so here we get to a slight problem. So we've said LK minus LK naught for the substrate is minus 19. That for the product, minus 14. But the problem is we don't know how much linkage has gone into that small circle. So if we were just to assume for the sake of convenience that it's probably about minus 1, we would get a delta LK of plus 4. But I'm not going to ask you to do that. I'm going to tell you a slightly different approach that we took to try and get around this problem. If I'm doing okay for time. And that was to instead start with the substrate, which has 2 perfectly evenly spaced recombination sites in it. So recombination makes 2 circles which are almost identical. That's fine. So the only difference between these 2 circles is that this one has maybe 1 or 2 base pair changes in it that introduces a restriction site that allows us to cut 1 circle away from the catenate. Okay, so again, we get to run this on a 2D gel, and what we get then is, mark them again to help you count them the way I want to count them. So in red, this is a ladder of produced from the substrate plasmid, this 2.4 kb plasmid, and we can see individual topoisomers that differ, again, each one from the next by 1 unit of linkage. And this now is a ladder made from the circular product of recombination, this 1.2 kb circle. And again, we can see individual bands. And so this is the slowest band, so it's 0, minus 1, no, actually, I think because of the way I've run this gel, things get more positive as we go round clockwise here. So this is actually 0, plus 1, plus 2, plus 3, plus 4. This is 0, plus 1, plus 2, plus 3, plus 4. So we start with a single topoisomer again. So we purified a single topoisomer and run it next to the ladder. And you can see, I mean, it's slightly contaminated, but almost all of the starting topoisomer is at this position, migrating at the same position as this band in our topoisomer ladder. And the products we get are, so there's not actually now this time a single product. We get a distribution of products with different amounts of supercoiling, and they are over here. We would expect this to be roughly a Gaussian distribution with, I'll show you the original gel in a minute so you can see whether my annotation, how accurate you think my annotations are. But the center of the distribution of products that we get is at this position here. And so let's just look at this without the bias of my nice bright yellow lines. So this is the topoisomer we started with. This is the main topoisomer product, but I think there are 2 faint bands here and here that are the topoisomers on either side of that. And then, well, let's just try counting it here. If we say this is 0, this is plus 1, plus 2, plus 3, plus 4. So the topoisomer we started with is plus 4, counting from this 0 anyway. And the product we get, this is 0, plus 1, plus 2, plus 3. The brightest one there is, sorry, the one at the top here is plus 4, which corresponds to the brightest one in the product of a recombination reaction. So that would give us 2 times plus 4 for the product, minus plus 4 for the substrate, a linkage change of plus 4. And you can actually count from 0, another 0 in the gel, which is this 0 in it that's at a different concentration of intercalator up here, and you get the same answer, round about +4 for the linkage change of the recombination reaction. So that means, oops, that's my timer. That means the linkage change we see is consistent with this holiday junction model for the strand exchange, which fits with what everyone thinks because XERC and XERD are tyrosine recombinases, and there are, it's well established that these tyrosine recombinases go through a holiday junction intermediate in the recombination, and there are crystal structures of, for instance, the Cree recombinase, which is another tyrosine recombinase, complex to a holiday junction that looks very much like my little cartoon here. And I think I'm just going to wrap it up there. So thank you for listening. Thanks to John Bath and Claudio Len, who started a lot of this topology work in Oxford with me, and people who carried on the work, mainly about how Pepe binds to DNA. Sarah Gurley and Martin Rines did a lot of the mutagenesis that allowed us to make this model. And also thanks to Marielle and DeWitt for their collaboration on the tangle analysis and future tangle analysis. Okay.
So I got a question. You had 3 topo isomers for the product, for the delta LK, and the max gives you delta LK of 4. And the other 2 give you delta LK of... Is that right? I mean, there were actually sort of 3 of them, and this was the biggest,
So on this one,
the brightest signal.
Okay. So let's actually look at this, see if you can see it on the gel. Does it show up here? I can see it on my screen here. So this is the brightest one.
And there are these other 2.
And these other 2 are fainter. But so the reason for that, if we go back to this cartoon, is that it's making 2 circles that are identical in size and are big enough to have, well, the linkage can segregate into the 2 product circles differently. So if this is -8 and the linkage change is +4, the -4 that's left, I'm making these numbers up as they were, could segregate -1 in this, -3 in that, -2, -2, or the other way around. And we just look at 1 circle, and so we see that distribution. Yeah.
So why didn't you measure the OK in the little circle? It's so much easier than your 2D gel analysis.
Because I didn't know how to. But
I didn't tell you exactly what
well, one of the problems is that you have to be confident that you're measuring the linkage in the small circle in the same conditions that you're measuring the linkage in the big circle, because of this 2 little assumption, well, effectively, because you're well, it amounts to, yeah, that what you're measuring is the difference between LK and LK naught, and LK naught can change depending on the conditions.
Absolutely.
So, but you could probably do it because the gel I showed you was done in the absence of any intercalator, and probably, I mean, salt will change LK naught a bit and things like that, but probably the errors will be small enough that we could do it and show that it was -1.
Steve? Yeah.
In your ligation experiment that led to the 5-2 knot,
Yeah.
what's the proportion of product that's knotted versus unknotted?
It still seems to be the majority is unknotted in the way that experiment was done.
Have you quantitated it?
I don't, you see, these experiments were done a while back, but I don't remember actually quantitating it, no. But, well, I mean, so you might be getting almost 1-to-1 unknot to this 5 knot.
Yeah, there's quite a bit of it there. So that suggests that the short end, the end that's closest to the synapse, is probably pretty tightly held.
It's actually, yeah, and that it actually ends up ligating through this loop a
Right.
significant proportion of the
Right.
time.
Right. That's a remarkable result. I would not have expected that naively.
No, I didn't. So this experiment was done two ways. If you look at this, the sites I've shown you here are in inverted repeat. They're pointing towards the right. They're pointing away from each other. Let me check. I'm not telling you a lie. Yeah. No, they're in direct repeat. I was telling you. They're in direct repeat in this DNA molecule. The experiment was done another way then with an inverted repeat. And for that, you will get, that will form something like that. And when it was done that way, we got almost all knotted things. So very little of the unknotted circle, and a lot of the three-noded knot. But also, I think, if I remember rightly, it's significant. So if you just join that to that, you'll get a trefoil. But if this ends up going through the loop again before rejoining, you get a five-noded torus knot.
Right.
And I seem to remember we got quite a bit of that as well.
Well, for what it's worth, I think we can now predict the distribution of products that you see there based on statistical mechanics.
So and the other thing, of course, you can vary is the length of these different arms. So just because of convenient restriction sites, this one ended up being quite short and that one ended up being quite long, but we could vary that and presumably get different products.
I'm happy we are having discussions. So, Marek, I saw you from very beginning. So, one short question.
I think it's a related question. Is anything known about the energetics of these systems? For instance, if you have a reaction in which a 3300 base pairs went into 330, okay, so what is the energy balance? And the related question is the distribution of this topological, the distribution of these various topological objects, has anything to do with temperature?
So I've not done any experiments like that or calculations like that. A significant driving force of this reaction would be this change in supercoiling. So you are losing effectively 4 negative supercoils and turning them into these relatively loose.
But what is the energy
catenations.
difference between what do you gain or lose?
Somebody else is going to have to do that calculation for me. But that would be interesting, yeah.
Okay, so thank you once more.